In this video, we're going to learn about DNA replication. So we'll learn how certain enzymes are involved in creating an identical copy of DNA so that the cell can divide. First, we'll take a look at the two different theories for replication that existed shortly after the discovery of the molecule. So the first one was conservative replication, and in this model, the two strands of DNA don't unwind, and from this, a brand new copy of DNA is produced and the original molecule remains intact. So if you look at this diagram, you see your original molecule, and then over time you end up with two molecules. One of them identical to the original, so it's outlined here in purple, and then one completely new strand. The way I like to look at it is as if you took a photocopy of the original molecule. So you didn't alter the original at all, and you ended up with a complete copy. Now, the second model of replication is the semi-conservative replication model. And in this one, you start off with your original molecule again, so here it is. But this time, the parent molecule unzips, so it opens up. And then each strand acts as a template for replication. So you end up having two molecules being built from that original template. And in this molecule, or this model, you end up with two new molecules, but each of those molecules has half of the original DNA and half of the new DNA, or the newly built DNA. And this is the model that we hold to be true today. So there are several enzymes and several steps involved in the process of replication, and it kind of makes sense because DNA is an extremely long molecule that contains all of the genetic information for the organism, so you want to get the steps correct. So there's going to be a lot of players involved. So first of all, the strands of DNA need to be unwound. We'll talk about the enzymes involved in that in a little bit. Then a primer has to be added to the DNA parent strands, and then an enzyme binds to build the copies of the DNA that will be the new molecules. And then finally, the primer needs to be removed, and then the strands need to be all glued together so that everything is ready to be packaged for the next cell. The first thing that has to happen is the strands of DNA need to be separated or unwound. So there are two enzymes involved and then some proteins that help stabilize everything. First of all, DNA gyrase, which is found right here, it's the little pink molecule, helps unwind the DNA and prevents negative supercoiling, so you don't want it to get all twisted, so it prevents that twisting. Then helicase binds as well, and that's shown like this little donut here, this purple donut, and it unzips the strands and holds those strands open and prevents them from closing in on themselves. But it needs a little bit of help. So what happens are these single-stranded binding proteins join up to the backbone of the DNA molecule, and it prevents the strands from combining back together or rejoining or hydrogen bonding back together. Now the next step is a little bit strange because the enzyme that builds DNA, which is called DNA polymerase, can't actually bind to DNA. It can only bind to a primer, and that primer is actually made out of RNA. So the first step that happens is RNA primase, also sometimes called DNA primase, adds a small primer of RNA to the 3' end of the DNA. And this primer will be removed later after the whole process has taken place. And the primer is complementary to the parent strand. So if you take a look here at this drawing down here, we have our original strand, and it's running from 3' to 5'. And its code is ACTGA. So a primer comes, primase then builds a primer going from 5' to 3'. That's the only direction these enzymes can work. And it's complementary to the parent. So across from the A is a T, across from the C is a G, across from the T is an A, and so on. And again, it builds from 5' to 3'. Once the primer has been added, DNA polymerase 3 binds to the primer and continues building the chain, or building the molecule. And DNA polymerase 3 adds complementary bases to the parent strand, and the strand, again, as said before, is built from 5' to 3'. So if you take a look here, here's our original molecule running from 3' to 5'. And then here's our primer that goes here. And then DNA polymerase III can bind to that primer and continue the process. So across from C is G, and then GC, AT, TA, and so on. And it continues until it gets to the end of the DNA molecule, and then replication will have stopped. Once the DNA molecule has been copied, the primers now need to be removed. And the enzyme that's responsible for that is called DNA polymerase I. Now you might be wondering where DNA polymerase II is. There actually isn't one. And DNA polymerase I removes the RNA primer and replaces that primer with DNA. So you can see that happening in this diagram from BioNinja. So this was the primer, it's erasing it and then bringing in DNA nucleotides to replace that original RNA primer. Once these primers have been removed, we need to glue those sections back together, and those sections are glued together by an enzyme named DNA ligase. So ligase kind of is the glue that holds everything together, and that is the end of replication. Everything has been complete, and we now have two identical copies. Now, you might have been thinking of this already, but DNA replication can only occur in one direction, as I've mentioned before. It can only be built from 5' to 3'. But if you remember, DNA molecules are anti-parallel to one another. So that means that there's going to be a leading strand where you can build the molecule continuously, and then there's going to be a problematic strand that can only be built in piece by piece by piece format. As the DNA continues to unwind, you have to add a new primer, build it, add another primer and build some more, and so on. So I'll illustrate that for you. So if you take a look here, we have our DNA molecule. It's begun to be unwound, so helicases unzip the strands. And we have one molecule that can be built continuously. So we put our little primer in, and then we can build the leading strand continuously because it's being built in the 5' to 3' direction. So everything's fine. Now the other one, it unwinds a little bit. We can add a primer, and then we can build with DNA polymerase. When the strand unwinds a little bit more, you can add another primer, and then you fill it in with more DNA, and then that has to happen again and again throughout the whole stage of replication. So the one that's on the left, this one right here, it's called the leading strand. And the one over here is called the lagging strand. Each of these little pieces that we see here have a special name, and they're called Okazaki fragments, named after the scientists, the husband and wife scientists that discovered these fragments. So the lagging strand, not so convenient because it has to happen in piece by piece format, but the leading strand can continuously be built. So this image from BioNinja shows everything happening all at once. So again, we'll just highlight everything. So the first thing that happens is DNA gyrase and helicase and the single-stranded binding proteins all combine to the DNA to help open up the strands and unwind it so that you can begin the replication. Over here, this is the leading strand. So this has been going on for quite some time. So this is moving on up as it's unwinding more and more. So the leading strand is built continuously, but the lagging strand has to happen bit by bit. So again, remember these little bits are called Okazaki fragments. And everywhere where we have to remove a primer after DNA polymerase III has done its job, DNA polymerase I has to come in and remove the primer, replace it with DNA, and then DNA ligase joins those fragments together. So you would only need to remove one primer over here and a whole bunch of primers here because we have to do this in pieces because it's the lagging strand.